Hello students, welcome back to my channel Competitive Botany. In this video, I am going to give you a brief lecture on linkage maps. So, we are already discussed in the previous sessions what is linkage and what is crossing over. So, you need to understand crossing over and linkage phenomenons before going to see the linkage maps. So, discovery of linkage that reveals so many facts. So they are nothing but, so let's see one by one. Many genes, they are located on single chromosome. This was revealed by a phenomenon linkage. Many genes, they were placed on a single chromosome and they are present in a linear manner. They are arranged one by one. They are arranged in a linear manner. As well as the genes present on single chromosome, they inherit together to the next generation as a linkage group. So they inherited to the next generation as a linkage group. Okay. So the number of linked groups in any organism is equal to their gametic number. It is equal to their gametic number. That means, for example, in humans, there are 46 chromosomes present in every somatic cell. Okay. So, their gametic number is 23 chromosomes. That means all human individuals are having 23 linked groups or linkage groups. Okay, so why? Because we have 23 pairs of homologous chromosomes. Okay, so homologous chromosomes are having same gene content. That's why we are having 23 linked groups in our body, in our every cell. Okay, we are already talked about a phenomenon crossing over. Okay, so linked groups, these linked groups, they are separated by a phenomenon called as crossing over. Okay, so linked groups were separated by a phenomenon called as crossing over. So each gene has specific order and location on the chromosome. Every gene has specific location on the chromosome and they have specific order. They are arranged in a specific manner. That means, for example, A gene, after A, B gene is there, after B, G or C gene is there. Okay, so this is the specific sequence of genes. They are arranged on chromosome in a linear manner. We should remember. Okay. Based on these observations, a scientist known as Sturdy went. So he is the student of Morgan actually. So he developed an idea that is nothing but the percentage of crossing over could be used as a tool to determine the positions of genes on the chromosomes. Okay, he considered all the observations. So just we have discussed. So he considered all of them. He has an idea to determine the relative distance between genes depending upon these observations. So depending upon the percentage of crossing over, we should we are able to calculate the distance between genes. So that was proposed by the scientist Sturtevant, who is the student of Morgan. So he given the first chromosome map of Drosophila. Drosophila melanogaster. Okay, he has given for the first time chromosome map of Drosophila. Okay, so he reported 5 genes present on X chromosome of Drosophila. X chromosome of Drosophila. So, he has represented a graphical representation. He has given a graphical representation of those 5 genes on Drosophila X chromosome. So, the genes are arranged in a linear manner. The genes are arranged in a linear manner. So, this graphical representation of genes on a single chromosome is known as genetic map. Genetic map or chromosome map. Or chromosome map. So, the distance between two neighboring genes. The distance between two neighboring genes is proportional to the frequency of recombination in between them. So, the distance between these two genes is proportional to the frequency of recombination is in between them. Okay. So, the units used in the mapping are called as centimorgan or map units. Centimorgan or map units. Okay. So, let's see the construction of chromosome maps. Let's see the construction of chromosome maps. Let's see first 2-point test graphs. So, you need to know what is 2-point test graphs. So, if we take 2 genes at a time to observe its recombination frequency. If you take 2 genes at a time to observe its recombination frequency, then it is called 2-point test graphs. So, this is done for knowing the distance between 2 genes. It is done for knowing the distance between 2 genes. So, that is called 2-point test graphs. Let's see 2-point test graphs. Okay. So, for example, in Drosophila, vestigial wings and black body color genes. So, in Drosophila, we are going to see the distance between these 2 genes, vestigial wings and black body color. We are going to see the distance between them. Okay. For this, we need to cross a wild variety with a mutant variety. Okay. We need to cross a wild variety with a mutant variety. Let's see the cross. So, here we are going to cross a wild variety with a mutant variety. Okay. So, let's see the crossing. Okay. So, from this we get 1. From this we get 1 gamete each. Okay. So, this is the wild variety. From this we are getting this gamete. This is the mutant variety. From this we are getting this gamete. By the fusion of these two, we are getting a F1 hybrid. Okay. So, this F1 hybrid is having both the wild chromosome as well as mutant chromosome. Okay. We need to do test cross this F1 with a double recessive parent. We are going to do a test cross. So, now we are doing a test cross. Test cross is nothing but F1 individual is back crossed with its recessive parent, that is called as test cross. Okay. So, here we are getting the gametes in this way. So, this wild chromosome will coming into 1 gamete. This is coming into 1 gamete. After this, recombination occurs in between them, and from this 1 gamete will come. They are going to fuse randomly for getting of offsprings. Let's see. These are the gametes are produced from the F1 as well as recessive parent. Okay, from the recessive parent only we get one gamete because they are having same gene content. Okay, so here we get one wild chromosome and one mutant chromosome, and by the recombination in between these two we get two gametes. So let's see this one, these gametes. So this is Vg plus and this is B plus. Okay, so by the recombination Vg plus B first gamete, Vg B plus second gamete, by the recombination we are getting two gametes. So they are fused with this gamete. These four gametes will going to fuse with this gamete. We are going to get offsprings. Let's see. So we got four progeny. So by the fusion of this one and this, we get this one. By the fusion of this one and this, by the fusion of this one and this, by the fusion of these two gametes, we get this. Okay, these are the four types of progeny obtained. Okay, so here we are going to see how many progeny are obtained in the F2. Let's see. So these two are the parental combinations. So these two are the parents. So parents are obtained in more number. So let's see. Parents are obtained 405. So these are obtained 415. These are obtained 90 to 88. Total progeny are obtained are 1000. Among 1000, so 405, 415 are parental combinations. So these two are the recombinant combinations. Okay. So depending upon these values, we are going to get frequency of recombination. Let's see the frequency of recombination. So to calculate the frequency of recombination, we know the equation. So that is frequency of recombination is equal to number of recombinants produced in the test grass by total progeny of the test grass into 100. So simply we can calculate this by substituting the values. So let's see. So number of recombinants were produced 92 plus 88. These are the recombinants, these are the parental. We can calculate later parental. Let's see first recombinants. So 180 by 1000 into 100. So simply we get 18. So that is nothing but 18 centimorgans or map units. Okay, so now we are going to calculate the frequency of parental combinations. So frequency of parental combinations is equal to number of parental combinations produced in the progeny by total number of progeny into 100. Okay, so number of parental combinations produced are 820. That means 405 plus 415 is equal to 820 by total progeny is equal to 1000 into 100. The answer is 82. 82 centimorgans are map units. Okay. So now we are going to calculate the distance between vestigial wings and black body colored genes in Drosophila. For this we need to calculate the distance depending upon the equation, that is number of crossovers in parental progeny into frequency of recombination in parental progeny plus number of crossing overs in recombinants into frequency of recombination. So let's see, by substituting these values we can get the distance between these two genes. So here I am going to, so in parental progeny crossing overs are 0 into parental recombination is 82. We are already calculated that. Plus number of recombination, number of crossing overs in recombinants is 1 crossing over is occurs in between those two genes. So into recombination frequency is equal to 18. Okay, so by multiplying this, it is going to be 0 plus 18. So that is equal to, the answer is 18 centimorgans or 18 map units is the correct answer. So now the two genes such as vestigial wings and black body color genes are separated by a distance 18 centimorgans or map units. So 1 morgan is equal to 100 centimorgans. So 1 centimorgan is equal to 1 map unit. You need to understand. Okay, in this way we can calculate the distance between two genes. This is about 2-point test graphs. So in the next class I am going to give you 3-point test graphs. Keep watching my channel. Thank you.